again, PIXUL, multi-sample Sonicator, in the agenda. So we're going to talk about principle and limitation of sonication, PIXUL sharing data, some ChIP-Seq data, conclusion Q&A, and then talk about the evaluation program and additional Q&A. So, PIXUL is very simple, very rapid, works in 96-fold plates, very easy to do, and consistent shearing of DNA, RNA, and chromatin, and even protein, whatever. So it's very consistent. It's a big point of the talk. So there is the machine, we've seen it already. Now, how does sonication work? So sonication, at least some of the Sonicator, the way it works is that there are transducers like sending ultrasounds, and these ultrasounds will be focused in the buffer where the DNA is inside the well, and there will be bubbles, and these bubbles can expand and then shrink, expand, shrink, expand, shrink, and at some point they're so big, they're so energetic that they collapse and they basically implode, and that releases a huge amount of energy. So there is like heat, there's going to be rust, so oxygen species, there's going to be shearing forces, shock wave. I wouldn't want to swim in this well, it's very rash, so for the chromatin and DNA. But it works very well, and that's how sonication works in general. So that's one bubble, cavitation bubble. So you can see that this bubble is formed and then gets bigger right inside the well. That's one bubble. There are millions of bubbles. And you can see that it shrinks, goes a bit bigger, and then shrink, a little bit bigger, shrink. It's not smooth, and at some point it implodes. And it's so energetic that this single bubble is producing light. What you see here is light emitted from one bubble, one cavitation bubble. So what it does, it induces heating, oxygen radicals, physical force, so there's a shock wave, and there's also shear stress of oxidation. And all these guys will induce shearing of the DNA, RNA, and chromatin. And so that's how the PIXUL works. So what is the bottleneck in general for sonication devices? And so we have some competitors, I'm not going to talk about them today, but we know them, and none of the competitors have 96-fold plates. It's individual tubes. Even some of them have sort of an array of tubes, but you need to buy individual tubes, probe in it, and they're very, very expensive. And we work with a $10 plate. It's relatively cheap. So, so that's the thing. And so basically, what happens is that people that use regular Sonicator, they work with cells in plates or in dishes, and then they need to manually transfer that into tubes to go into Sonicator. Then there are usually steps of degassing, cooling, adding water, cleaning, and a lot of things that we have to do before being ready to Sonicate. And that takes usually 45 minutes-ish. So that's another bottleneck. Third bottleneck is consistency. So if you guys did a lot of ChIP-Seq, you'll agree with me that you can do a ChIP-Seq, a sonication day 1, day 2, you do the same sonication, it doesn't work. And the only difference between the 2 days is that yesterday was sunny and 2 days straight. And you don't know what's happening. I mean, at some point we had a little god of sonication next to the Sonicator in my PhD lab, because you never know if it's going to work. So it is a big bottleneck. And then finally, well, if you have single tubes, you need to put that again in 96-fold plate if you want to work in 96-fold format. So these are all the bottlenecks with the regular Sonicator. Now, we just get rid of that by using simple, affordable, plastic sterile plates with round bottom, in which you can grow your cells directly in the plates. If you want to do a drug treatment, drug screening, you can treat your cells, and then put it in the Sonicator with the right buffer, hit stop, and it's good to go. So it's very easy, very straightforward. So we have, alleviate also these issues, much more consistency, no degassing, no filling with water or buffer. It stays there it can stay there for a while. You don't need to do anything, just turn it on, wait 10 minutes, cools down, and then it's good. So that's another thing. And then lastly, well, if you work in 96-well plate, you don't need to, you can do your IP in the 96-well plate. There's no transfer in the end. All right, so that's how the PIXUL works. So basically, you have your 96-well plate. Each plate has 96-well wells, and then between each well, there are transducers and graphite lens. And so what happens is that there are going to be an array of transducers. There are, I think, 24 transducers. Yes. 24. And so basically, what happens is that there are so many transducers that you can almost, not individually, but you can select each parameters that are different for each of the columns of 12 columns of the next 12. So basically, you can share DNA in, column 1, RNA, column 2, chromatin, column 3, different parameters. So that's also very easy. So transducer will basically shoot ultrasounds that will be focused through a lens, and that will produce cavitation bubbles that will in turn shear a nucleic acid. All right, also very intuitive touchscreen, also very Apple style. You can select the columns here. I don't work for Apple, so I don't have anything there. But it's very minimalist, I would say. Let's put it this way. And so 12 columns, just select the columns you want, and then parameters, hit start, and it's good to go. So very, very easy to work with. Inside the Sonicator, once you open the lid, there is a gasket where you can safely place the plate, and then seal it, shut it, and activate the sonication. All right, so what can you do with all these, with this machine? So you can sonicate DNA, either directly purify with a phenochloroform or a kit or whatever, and you can also sonicate DNA directly from cells. So you can grow your cells, don't need to do phenochloroform extraction, throw them in the plate that will give you. You just PBS, tap it, remove PBS, add the buffer, the plate in the Sonicator, and sonicate. It's as easy as that. The extraction is done, sonication is done, then you just need to purify your DNA, right? Get rid of the thing that you don't want. And we have a nice kit that will, arrive soon that will be able to do that. So it's very, very, very easy to do if you want to do that in batch with 96 different samples. So that's one thing that we can do. Chromatin sonication for ChIP-Seq, high-C, sonication of purified RNA for epigenomics, people that want to map metal adenosine, 5NC, 5HMC, picopoison. You can sonicate RNA as well. And we get usually a size between 200 and 600 listeners. Consistency. So, okay, some results now. The first thing I wanted to mention, and it's very, very, very important, and whatever sonicator, never use water as a diluent if you want to sonicate DNA. If you do that, there's going to be heat, as I said, and no ions. And so there will be negative charge that will oppose each other, and so you will end up with single-strand DNA. It will be very, very beautifully sonicated, very beautiful, but it will be single-strand. Here's the different buffers that we were using to test the sonication with salmon sperm DNA. And as you can appreciate, we have a nice sonication with trace TE, if you want to go a little bit of EDTA, and water we don't see anything because it's a double-strand DNA chain. It doesn't capture double-strand DNA. If you run this on an RNA chain, you'll see the opposite. You'll see one nice lane here and blank here. And basically, and you can quantify as well with kits, what is the proportion of double-strand and single-strand. So my recommendation would be to use 3CHCl, pH 8, 10 millimolar, and you're good with that. Okay, so get consistent shearing with purified genomic DNA. Also tested with directed from cells. It's good too. And so it works with a broad range of DNA concentration to start with. We can work with 20 microgram to 0.5 microgram in one well. So you can do a decent amount of fragmentation. All right, so what about the sizes? Sizes here ranges from I would say 100 to 100 base pair to 700. Same here, and it's independent of the DNA concentration, of DNA mass, and almost independent of the time. So we recommend people to use 36 minutes, I think, 36 minutes sonication. Here's 30 minutes, but if you look at 10 minutes, it's a little shifted towards the high molecular weight, but it's very decent already. So you don't have to. I mean, we recommend basically to do 36 minutes, but it's very, as you can see, it's very robust. So that's something that is nice to do. It's very, you can reach these results quite quickly. So yeah, DNA concentration does not affect sonication. 30-minute sonication gives you a profile of 200 to 700 base pair. So as I said, there is a way to avoid phenylchloroform extraction and just sonicate cells with the right buffer and extract the DNA after we beat. And so we did the experiment too, and you can see that, well, if you start with 100,000 cells or a million cells, you're going to have a different amount of DNA. The ratio 260 to 80 are super clean. I actually have better ratio with that than with the phenylchloroform. Usually you have more towards 1.89. Here it's really 1.81 or 1.82, something. It's super, super, super rapid. So no contamination of proteins, no contamination with RNA, and very clean signal. Average size is again around 300, 400-ish base pair in this experiment, so suitable for cool down in sequencing. And everything is double-stranded, so there's no issue with the library prep in the upstream experiments. So, same message as for the purified genomic DNA. It's very easy to use with cells directly ground in the plate. And that's the consistency results I wanted to show you. So we took salmon sperm DNA, diluted in tris, and then put that in different walls of the 96-well plate and just sonicate it. And as you can see, there is a very high consistency in sonications. It's very, very high. It goes from 75 base pair here to like, I don't think I can read it. I think it's 700, but you don't have anything above 700. And these are the two unsonicated DNA control. So very, very robust and good reproducibility. So that's good. And then next, people are interested in RNA modification. Personally, I am interested in RNA modifications. And so I insisted that we set up also the system for RNA. And so you can do sonication, can sonicate RNA. Usually people use zinc to do that, but then you need to purify your RNA. So you can just sonicate RNA in water, just plain water, with our plates, and you're gonna have fairly good results. Here you can see it's unsonicated RNA control, it's total RNA. So as you can appreciate, there are two RNA bands and then the tRNA band, and the sonication is fairly consistent between the three buffers, but looks a little bit better with water. So I would just recommend you to use plain water, and RNA-free water, of course, and then you should be good to go with that. That's for RNA, and finally chromatin. So we did extensive testing on chromatin, which is the most challenging, structure of DNA, and it can be finicky. So we worked a lot on that, and so we have now a range, I think, from 10K cells to 5 million cells per well. Okay, so you can sonicate from 10K to 5 million per well. Again, ground or transfer from the plate. Sonication is quite reproducible. It's it's about 300 base pair in this experiment, and get a little bit of peak here that we also observe with other Sonicator, and I do think it's heterochromatin that is very refractory to sonication. It would actually be interesting to sequence that. And these are unsonicated cells. So that's for the chromatin. And now some genome-wide data, right? Because we're doing ChIP-Seq or DeepSeq, and we want genome-wide data. So I will talk about 3 marks: K4 trimethyl, K27 acyl, and K36 trimethyl. So let's start with the easy one. So K4 trimethyl, it's a very good antibody. It's easy to chip, and it's one of the goals. You know, it's one thing that one wants to try when you use a new instrument. And so basically, we did K4 trimethyl in 293 T cells. We did an input and 2 replicates, and as you can appreciate, they're very much alike. Peaks are very sharp, very defined, and when we zoom into peaks, they're also again very sharp. Here, I don't know why the gene didn't appear, but it's right at the TSS, which makes sense because it's a H3K4 that is enriching in promoters. So some genome-wide analysis, you can see here a metagene. Again, most of the signal is clustered around TSS, no signal in the input, and again, very, very consistent results between the 2 replicates. This is the aggregate, and these are the heat maps. Next, we looked at the overlap, and there is a very good overlap between the 2 experiments. That is very high significant. But I wanted to show you a heat map version as well because it's very binary here. And the way I did is basically using a peak file from one of the replicates that I chose, was replicate 1, and then plug the big weak signal from the input, that with the negative control, the replicate 1 that's positive control because you basically compare it to itself. So that's the best it can be, and then that's the query, which is the replicate 2. And as you can see, it's very much alike. And actually, I actually looked at it earlier today. If you zoom in, you can see some lines that are, some patterns that you can see by eye. It's actually nice to do that. So, lastly, FRIP scores. FRIP score is a measure of efficiency of the chip, so it measures the amount of reads, or the fraction of reads that fall into peaks. So the more FRIP score is high, the more reads you have in peaks, and outside peaks you don't have any reads, or barely any reads. So ENCODE set a threshold at 1 or above 1, but we want it to be much higher. So here with K4 trimethyl we'll have a FRIP score of 23-ish, which is pretty decent. Finally, we compared to ENCODE data, published ENCODE data. You can see here that the peak co-localized very well, and I did the same type of representation with a heat map, using as a peak file the ENCODE data, and then plotting the signal from input, replicate 1 and replicate 2, and you can see that there is a nice correlation at the center. All right, K27 acetol. So personally, I don't have a huge experience with this one, but I find it a little dirtier in K4 trimethyl. It's still very clean, but not as cute as K4 trimethyl. So we wanted to do that too. We did the same type of analysis, and as you can see, the patterns are exactly the same. The shape, if you were to draw a line here like that, and then superimpose these lines, they would be completely overlapping. So very, very robust. If you zoom into peak, you can see a very nicely defined peak at the TSS as well. And now some genome-wide analysis, same thing. So a metagene analysis, you have cluster of reads at the TSS, a little bit upstream as well, and nothing downstream, or very few downstream, which is expected. Second, the overlap, the consistency of the results. We do get a very good consistency. And when you plot this as a heat map to have a more quantitative value, you can see that the query is as good as the positive control. It's very reproducible. Finally, the FRIP score, again set by ENCODE, it's above 1, but we get 25 here. We also compared K27 cell to ENCODE data, and you can appreciate that. You can see that it's a little dirtier in general than K4. I mean, it's valid for everything. I think this mark is a little risky, but it's nevertheless very good peaks and very nice correlation with public data set. And when you plot the vet file as a heat map, you can see a very nice correlation. All right, let's go back to the bad guy. So K36 trimethyl, it's a kind of nasty chip to do. It's very broad. It's a little bit more dirtier. And so personally, I'm trying to stay away of these broad marks. But we wanted to test it because one has to be able to ship anything with that and not only easy stuff. So we did ChIP-Seq with K36 trimethyl and also K9 trimethyl, which I won't show you, and get good data. So, all right, so here you can see that it's again a little bit dirtier, but you can clearly see that you have, and it's expected, you can clearly see that you have peaks, and these peaks co-localize with the gene bodies of active genes. When you zoom in to certain peaks, you can see that it's very well enriched inside the gene body and depleted outside. And if you overlay, I didn't do it, but if you overlay K9 trimethyl, K27 trimethyl, you see a negative correlation. So you will have like a large lump here, and then a dip, and then a lump here. And it's really anti-correlated. It's actually quite cute. So some genome-wide analysis. First of all, we get the exact same profile we should get, meaning depleted at the TSS and then rising up towards the 3' end of the gene and then going down again. And it makes sense because that's what has been published for a while. As with the overlap, we have also a very good overlap between the two experiments that is highly significant. And finally, the Fruit score, which is a measure of the ChIP threshold set by encode again at 1, and we want it to be much above that, and we are near that for 30, so it's a good Fruit score for that type of chip. So, in conclusions, we have consistent, and that's very important, we have very consistent results. It's fast, it's efficient, and it's affordable. Right? You can sonicate chromatin, DNA directly from cells, DNA purified, or even RNA or proteins or whatever you want to sonicate. It's 96-fold plate, very easy to use, very straightforward, but very few buffers, very few purification steps. Target size range for most of the applications between 200 and 600 base pairs, so suitable for IPs and library prep. And so that you can basically target the epigenome or the epitranscriptome. And with that, I'd like to thank Nate, who's sitting here, who's done, I would say, 85% of the wet lab work on that at Active, something like that. So he's done the bulk of the job. I was sitting behind my computer. So he's done a super great job. We wouldn't be able to do that if he weren't there. So big thanks to him. That's thanks to him that and other people. And then also, he's not the only one, right? Adam, that was my predecessor at Active. I joined 6 months ago, so I inherited this project. So big thanks to him too. Eddie, that is the VP of R&D, and he's been always very supportive and very patient and great to talk with. Ted, the CEO, that was also very patient, and I know you're looking now and you must be smiling. So that's that. Joe, the founder, is super excited about the PIXUL, coming down to the lab with many ideas. Why don't we try that and that and that and that and that? I'm like, okay, Joe, just a minute. He's very excited. He's a very excited guy, and it's awesome to work with him too. So a big thanks to Nate and all these guys, and of course everyone at Active that participated in this project. And it's a team effort, right? It's really a team effort. Now, from our collaborators at Matchstick in Seattle, we want to thank Tom and also the lab of Carol that helped us a lot with the settings and preliminary experiments. And with that, I think I'm done and I can take any questions.
Yep. Thanks. So I'm really curious regarding variation between sequencers. Did you try a couple of things? One thing that I've missed is did you try doing some even scatter plots to see if you have some regions of the genomes that differ?
Yeah, we do have scatter plots as well. Yeah, it's already a long presentation. I couldn't put them, but we do have them.
And did you try to take exactly the same sample and split it between two different sonicators?
So yeah, so we have done. So there is. You can see that in NAR paper that there's an NAR that was published. I believe, a year ago, and that deals with that. So they have done comparison with PIXUL, with Gavaris, I think Gavaris and Digenodes. So they have comparisons there.
Yeah, so for DNA sonication, so you said that DNA sonication.
DNA, yes.
DNA sonication. So you said the target is around 200 to 600 base, right?
Yeah.
If we change the power or timing, can we set the target to some other sizes, for example, like 100, 200, 300, or something?
100, I don't think you'll reach that. I don't think you can reach that with any Sonicator. It's like a spaghetti. It's easier to make a long spaghetti than a short one.
Yeah.
So it's going to be challenging to get that. But you can have bigger sizes if you want. You can Sonicate less. Yeah.
I'm asking because, for example, like whole virus, they provide different protocols for different size, target size in the center, like from 200 away up to like
that is very true. And the 96-well delay from Gavaris, if you have to build it yourself, is going to be $500. Ours is 10.
Yeah. Okay.
It's a big difference.
And one more question for the 96-well plate. If I only use part of the plate, I can keep the rest.
No, we don't recommend to do that.
Okay.
Because the plate is hit by ultrasound, and it can damage the plastic over time.
Okay.
I wouldn't freeze it. I wouldn't keep it. I would just grow yourself there, do your experiment, and then throw it away.
So basically, the plate is only for one-time use.
Yep.
No matter how many samples you have.
But again, these are cheaper than one tube.
Yeah, of course.
So.
You tried only cells, how about tissues?
Sorry?
Tissues, have you tried chromatin?
We have did test on tissues, yes, and I believe it's in the NAR paper too. Yeah.
Yeah, and also for the
I can actually send. I should have put a link. Maybe we can send a link for the paper for the guest.
And one more question for each well. What's the maximum number of cells for like tissues?
It depends what application you're doing.
For example, like ChIP-Seq.
ChIP-Seq, 5 millions per well. 5 millions per well.
5 million. Okay.
Yeah.
But the protocol will be very similar, or time?
No, it will be similar.
Okay.
Just the dilution in the end that might be different.
What is the volume in each well?
Technically from 50 to 200. I wouldn't change. I wouldn't go below or higher. I think we're doing 100 now. That's the consensus, and that's what we recommend. 100 microliter. Yep.
What is the earlier versions used like a sticky tape on top, and when hitting on the sonication buffer for ChIP, for example, it would basically dissolve the adhesive.
Oh, I see what you mean. We don't have that anymore.
Okay.
I know you had mentioned
have
to
you
add.
tried different buffers with different detergents? Like what's the
Yeah.
detergents used?
Did we do that? Did we try, for example, with more SDS to see if the seal still sticks to it?
I have not tried a wide range of detergents. It's basically just
our detergent, yeah.
Okay,
But
what is that?
didn't you try it?
Yeah, and that's the
Oh,
top
that's
tape
what happened?
And then we use some like rubber
Well,
covers that
yeah.
kind of go into the wells to stop it from getting up to the top.
But do you have more contamination with that? Cross contamination?
Well, it's tricky. You have to make sure you spin it clean so you don't bring it up. Yeah.
No, we didn't see any issues with the current protocol, current machine, and current regions with the seals.
So currently the seals are this plastic adhesive?
Yeah, it's like qPCR seals.
Do you recommend a specific buffer, for example, for ChIP or reserve?
Yes, I would recommend our buffer. I already gave you the recipe for DNA and RNA. Come on.
But there's just one buffer for ChIP.
It is,
Or
as
you
for
don't have like
now, we
four
have.
buffers, different buffers. or
No. For now, we have one protocol, one buffer, one kit that we're working on.
So for chromatin shearing, there's one buffer, and then you add an additional buffer for ChIP prior to the ChIP.
Oh, Well, no, you need to dilute it. You need to dilute in the chip buffer. You can't do. I don't think we do the pull down with the sonication buffer. It has to be diluted in the chip buffer. That is provided in the kit.
And for the sonication directly on the cells, do you mean only suspension cells or even the cell still attaching to the?
You can do both.
Okay.
Yeah.
So you do not need to trypsinize the cells.
Don't go higher than. So I've tried 100,000 million, 5 millions. The 5 millions was super blobby and it didn't sonicate well. So I wouldn't go higher than 1 million per well.
Okay. So
If you want to directly extract unfixed cells.
okay.
Okay.
All right. Give it to you. All right, thank you, Ben.
Thank you.
So I'm Seth. If anybody doesn't know me yet, hopefully you will, especially if you decide to evaluate the PIXUL, you'll arrange for that engagement. So thanks a lot to Ben, and I'll get a credit. Thank you to Dave and the Center for Genomics for sponsoring us here to give this seminar, and also to allow you to evaluate the PIXUL instrument while it's in the Center for Epigenomics. So I just wanted to talk a little bit about how we can make that happen for you. So, first, this is the PIXUL. This is what it looks like. I think that showed you a picture. And the catalog number and the list price. Nobody fainted, that's good. Excellent, okay. So far, so good. And here is a list of the consumables that are available for you to use and for you to evaluate the instrument. So the 9612 plates, they're basic Corning plates. Like Ben alluded to, nothing fancy. They should cost between $5 and $10 each. So yeah, again, like Ben mentioned, it costs as much as just one sample on Covaris. So if you're getting the most out of the plates, this is 1% of the cost for using this compared to Covaris. The coupling buffer, this is not a one-time use, but the coupling buffer lasts for a long time. This is the buffer that sits in a well on the instrument. So there's a port to fill underneath the lid here to fill with the coupling buffer, and it takes half a liter of coupling buffer, and it doesn't need to be replaced very often. So that's consumable, but rarely would you need to change it. The Chromatin shearing kit, though, and chromatin input prep kits are available, as well as the gDNA shearing kit, which, like Ben alluded to, may not be quite as necessary because shearing just naked DNA is easier. And then also we'll be introducing a high-throughput chip kit shortly, and this takes full advantage of the throughput capabilities of the PIXUL instrument. So how do you evaluate it if you want to? So it'll be here until the end of the month, and we'd like you, if possible, to schedule with me. So just send me an email. We'll find a convenient time for you, and so I can help you get set up with the reagents that you need and so on. And we'll block out 1 hour time in increments. It takes about 30 minutes, 36 minutes to perform a run. If you need more time, no problem, we can arrange that. But we'd like you to have the opportunity to try it a couple of times if you like. So to give it a whirl, look at the results, evaluate it, talk with us about your results. We're happy to consult with you on how well it worked and so on. And then we'd like you to have a chance to try it again. If you want to change any of the parameters or look at different cell types or different assays, what have you. So if possible, let's get your first run scheduled for the next 1 or 2 weeks so that you have time before the end of the month to try it again if you like. So yeah, and we'll provide the consumables that you need, and then, but what we ask of you is feedback. So this is the whole point of our working with you guys, is to develop feedback. So to hear your thoughts on the instrument, how well it works, how well it sonicates, how well the results from your downstream assays, your NGS, your ChIP-Seq, how well it all looks. We really want to hear all that, and we're happy to work with you for optimization and for any changes, and we'll definitely work with you through the end of the month to generate all the data that you need. And then, let's see. So we do ask for the sanity of the Center for Epigenomics. So the instrument is in a dedicated room, so you won't bother anyone there using the instrument, but please do bring your own pipettes and pipette tips and so on. And you're welcome to contact me, of course, our tech support team, and or Ben, if you have any technical questions or any sort of logistical questions, or if anything comes up at all, and to provide your feedback. So that's basically it. Thank you for your participation today. Thanks again to the Center for Epigenomics. Thanks to Ben and the whole team. And any questions for us at this point about anything, the instrument or evaluating PIXUL? All right, so there is a quick start guide. We do have the manuals and everything available for you to review and for you to use for your evaluation. So I'll make sure that you all have the quick start guides and then access to the PDFs of all the other information that you need. And then if there's no questions, Dave, do you have any parting thoughts for the group?
Parting thoughts are thank you to you, Seth, and to Ben and the folks back at Motif for lunch and for the information and for letting us demo this PIXUL. Thank you guys all for coming. And if you have any feedback or questions, and as you evaluate and get results or want to hear our results, be in touch with me. That'll be great. Thanks everybody.
Thank you.